Kind of next question is a little bit similar to the last one, but a little bit different here. What are some of the biggest challenges in translating preclinical remyelination success to clinical benefit in patients? We've seen some success in preclinical studies, some animal model studies, some early stage studies, and things of that sorts. But how do we take some of that bench work and really kind of bring it to more clinical care and continue to advance it? Obviously, I'm not asking you guys to make a therapy right now, but what are ways in which we can continue to advance some of that research?
We should be asking us to make a therapy because that's our obligation. Like that's what we do with our day jobs. So that's exactly what we need. It relates back to what Paul said about biomarkers, and we need those biomarkers are a critical component of the translation, and they have to be assessed in the preclinical context. So we need to develop preclinical assessments that we can use that we can then directly translate not just the biology, not just the molecule or therapy that's used to afford remyelination, but also take measurement or to have assessments that we can identify or work on in the preclinical setting that we translate over into clinical practice. So I think that is probably one of the greatest, but also knowing when do we optimally remyelinate? Do we optimally remyelinate in the setting of inflammation or when inflammation has been is in the rearview mirror? Do we need to continuously remyelinate? Do we need to give drugs all the time, or do we need to give them in a pattern where we give them sometimes and then give people holidays or periods when they're not on therapy? Do we need to give it to people early in the course of their disease or later in the course of their disease? So those are all components that remain challenges for us. I think the challenges we are, at the threshold of answering. In fact, we have answered some of the fundamental ones, kind of as a add-on to the question you asked about biomarkers. With one of our clinical trials, with the Rebuild clinical trial, we actually showed subsequent to the assessment using electrophysiologic techniques that showed the effect of the drug clemastine for remyelination, that it not only had that effect in the visual system, but also on brain MR when looking at particular areas of the brain and outside of lesions, we could see evidence that there was a remyelinating effect. And on top of that, using neurofilament light chain as evidence, as direct evidence that even in short-term remyelination trials, we see evidence that there is protection of axons. So former postdoc from my lab, Ahmed Abdul Haq, did that work and established it in a paper published in JNNP just a couple of years ago. So really, this iterative process is critical in taking a wide array of drugs. We've been very happy with the work we've done with clemastine, but looking at drugs with multiple mechanisms of action and multiple different approaches, the kind of approaches Paul has taken, using different tools and techniques to show the same effect is also like having broad evidence that lots of different ways of remyelinating have the same effect on biomarkers. That's also a critical step for the field. It's similar to what happened with the immunomodulatory therapies, showing that using interferon beta and using glatiramer acetate both had similar biological effects, and then showing later that natalizumab had the same biological effect. Those were all really important steps in establishing the tools and approaches for not only assessing drugs' efficacy, but also monitoring patients once they went on those drugs.
And I think what I can say as far as thinking about some of the challenges translating preclinical remyelination success into clinical benefit is I think one thing is really the significant species differences that exist in the timing, the efficiency, and this capacity for oligodendrocyte regeneration and remyelination. So, I mean, for example, in mice, it takes around 3 days for a progenitor cell to form an oligodendrocyte, about a week for that oligodendrocyte to myelinate. Now, in humans, this takes at least 4 times longer, right? It's 14 to 21 days to form an oligodendrocyte, 4 to 8 weeks to form myelin in some cases. So, these species-specific differences are things that we need to be incorporating into our understanding of our preclinical models, right? And so I think this kind of tells us is that, there's a need to continue to incorporate, I think, current and emerging human preclinical models, such as brain organoids, iPS cell technology, as part of the discovery and development programs so that we can identify remyelinating therapies that have mechanisms that function effectively in the human context. I think the other challenge in sort of translating of preclinical success, which a number of us, including both Ari's team as well as my own, have had really strong positive data on the preclinical side for remyelinating therapeutics. And so you might say, well, why aren't these generating remyelination in patients right now? And I will say that we are in on the cusp of that with many of the trials ongoing and including some of those from Ari and colleagues. But I think the challenge is, which is also the opportunity, is this white space of remyelination trials, right? There's no currently approved remyelinating medicines for MS, right? And until we get there, we're going to have some uncertainty about, which of these preclinical models provide the best positive predictive value and clinical success. But I think, just to sort of say in closing on this point is, I think we are entering this era where we're going to see a lot of activity and success in the remyelination space. There's multiple ongoing or imminent clinical trials. And I think importantly, testing diverse mechanisms to drive remyelination in patients. And I think there's really room and necessity for multiple successful remyelinating therapies, as Ari pointed to earlier. And I think, as the saying goes, a rising tide lifts all boats. And I think we're going to be entering an era here where we're going to have a lot of excitement and success in the remyelination space.